Hello everyone. In our previous lecture, we have learned about how to retrieve the CDS and SNPs from the ensemble database. And today first we will learn to organize this data in tabulated form. As you are able to see here a table in which the first column is case number and second is SNP ID. How you may retrieve as many SNPs as you want from the database. I am just showing the three as an example, and you will designate a number, a case number to every SNP, like a SNP which is located at the 67th codon is numbered as 1, and the one which is at 129 position, which is a stop gain SNP, is documented in case number 2. This will make it easy for you to identify the SNP which you are talking about at a certain time, and also it will make it easy to document it in throughout your manuscript. So case number 1 has the SNP, the ID is given, and then there is consequence type, and then position of codon, then nucleotide chain in change in normal. You have GAA in normal gene, while in mutated you have GGA, and it is changing the amino acid from normal E to G. Now let me show you how will you construct the coding sequence using these SNPs. Let us start with the example of case 1. In case 1, the mutation is at the 67th codon. So first you will copy and paste the normal gene sequence which you have retrieved from the ensemble database. This is the case 1, for which first I have pasted the normal gene sequence, and now you will locate the specific codon which is at 67th position. For the convenience, I have already located it, and I have changed its single nucleotide. GAA has changed into GGA. So at the 67th position, which is, you will count the codons manually. Here is the mutation I have already incorporated in order to save the time, like GGA. And now you have constructed the CDS of the mutated protein for this missense mutation, which is located at 67th codon. While in second case, you can see here in case number 2, the SNP documented as stop gained, and it is located at the position 129, and in normal gene we have TGC, while in mutated gene we have TGA. So let us locate the 129. This is the normal sequence. Again, you will paste the normal gene sequence, and then we'll count the codons, and at the position number 129 you are able to see TGC, which will change into TGA. So now you will change the C into A, and hence you have created the CDS, or the coding sequence, for the mutated gene or the mutated protein, which will be formed in case of second SNP. While in third case, a frameshift deletion mutation is documented at 144 codon, and in normal gene we have CTG, and in which D is deleted in the mutated form, and CTG has changed into CD. Now let us locate 144. Again, you will first copy paste the normal CDS, and then here is the position 144. I have already counted it and labeled it now, or highlighted it, and it's CTG. Now you will delete the T, and after this the whole frame will be changed. Now definitely these are only two nucleotides. There are only two nucleotides left in this codon. Now you will make it three. You will make this codon to comprise in three nucleotides. You will incorporate in this codon the C from the next codon, and hence there is only CC in the next codon. Now you have incorporated the codon, the C from the next codon, and it has become triple C. In next, do the same practice, and hence the whole reading frame will be changed. As you know that the frame shift mutation changes the whole reading frame, so one deletion mutation has dramatically changed the CDS of our gene of interest, and so on. So this is all about the construction of mutated coding sequences using your SNPs. And now next task is to translate this nucleotide portion into the sequence of amino acids. For that, you might be familiar with the XPCE translate tool. XPCE is basically an online bioinformatics resource, and which provides you access to almost 160 databases and multiple online tools, and one of which is translate tool. So click on the translate tool. It will open a page, home page of XPCE translate tool, and you are able to see here a box in which you can paste your DNA or RNA sequence. So let us copy our sequence from, suppose, case 2, which is containing a stop gain mutation. Let us copy it and paste in the box. And now the genetic codes in the search bar where standard is written, this is by default, and leave it like this. Do not change it. And in output format, you can see there are different formats given, like in verbose methionine, the start codon and the stop codons will be labeled, and there will be spaces between the residues, while in the compact one, the methionine will be labeled with M and stop codon will be labeled with a hyphen, and there will be no spaces between the nucleotides, between the amino acids. Now it's up to you which format you want. Then click translate. It will give you multiple options for the protein sequence of your mutated CDS. Select the very first one in which you are able to see the protein sequence is starting from the methionine and it is ending at E, and then there is a stop codon. Because this was a stop gain mutation, so a stop codon was inserted at this place as a result of this mutation. Now copy and paste this sequence in the Word file and keep it for analyzing the effect of this SNP on the different attributes of your mutated protein. If you have designed, suppose if you have selected 100 SNPs, then you need to design 100 coding sequences, and you have to translate all those into the amino acid sequences. Thank you.